In the name of Allah, the most beneficent, the most merciful. First of all, I will introduce myself. I am Ghulam Rubani, student of BS Microbiology at UMT Lahore. Today I will discuss about transcription process. In transcription process, we will briefly discuss about definition, difference between replication and transcription, features of transcription, transcription in prokaryotes, transcription in eukaryotes, and finally we will discuss about reverse transcription. Let's start with the definition of transcription. Transcription is a cellular process in which messenger RNA transcript is synthesized by using DNA as a template. As messenger RNA is a type of RNA. RNA has three types: ribosomal RNA, transfer RNA, and messenger RNA. RNA is single-stranded and polymer of ribonucleotides. As a transcription is cellular process and it requires RNA polymerases enzyme. In below figure, you can see that this is double-stranded DNA and this is messenger RNA transcript, and this is RNA polymerase. This dimer is RNA polymerase. Now we will discuss difference between transcription and replication process. Many students got confused between these two processes. They think that these are similar, but they are quite different from each other. Here we will discuss about basic differences between transcription and replication. Transcription requires single strand in that specific part of that strand, but in replication we require both strands. In transcription, there is no need of primer, but in replication, we need primers. In transcription, RNA polymerases are used as enzyme, and DNA polymerases, helicases, DNA ligases are used as enzyme in replication process. Here is the diagrammatic chart of replication and transcription. You can see here is the replication fork. This is leading strand and this is lagging strand. You can see the primer in replication process, but in transcription process you will see there is no primer in this process. Now we will discuss about features of transcription briefly. Transcription is highly selective because there are specific signal embedded in the nucleotide sequence of DNA. And these sequences mark the start and end of DNA segment where transcription is to be done. And these signals give instruction to the enzyme that where transcription should start and where it's gonna be end. At start, many transcripts of messenger RNA are synthesized, and these transcripts are called primary transcripts. And a few of them are modified, trimmed, and converted into final and mature form of messenger RNA. And this final and mature messenger RNA is then transferred from nucleus to cytoplasm in different locations where protein synthesis is done. Now we will discuss about transcription in prokaryotes. Transcription consists of three main phases: initiation phase, elongation phase, and termination phase. In initiation phase of prokaryotes, RNA polymerase bind with the DNA double-stranded and the sigma factor create transcriptional bubble there. And in elongation phase, RNA polymerase add nucleotides and the synthesis of messenger RNA is start. In final and termination phase, when RNA polymerase reached the stop codon, we have studied three stop codon: UAA, UGA, and UAG. These are three stop codons, and one of them is present at termination site where transcription process will end. Transcription in eukaryotes is similar in prokaryotes, but it is more complicated because the RNA polymerase has three main different types in eukaryotes: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. And it also requires many transcriptional factor proteins, but in prokaryotes only one sigma factor is required. And in transcription in eukaryotes, we need upstream regulatory region along with promoters. And in this diagram, you will see that there are lots of transcriptional factor, and these are the types of RNA polymerase 2. You can see in series that and upstream regions, and all these will participate from initiation to termination, and finally we get a messenger RNA copy. Now we will discuss about reverse transcription. Reverse transcription is a special type of transcription. In this transcription, DNA is synthesized from single-stranded RNA. As we have studied retroviruses, RNA containing hereditary material class, they also contain a special type of enzyme called reverse transcriptase, and this enzyme play role in converting single-stranded RNA into double-stranded DNA. And when RNA retrovirus entered into a host cell, its genomic RNA will be transcribed into double-stranded DNA by the reverse transcriptase, and then this double-stranded DNA is integrated into host DNA. This process of conversion of single-stranded RNA to DNA is called reverse transcription. And in diagram, you will see that this single-stranded RNA is converted into DNA, and then this DNA is incorporated into host genome. Now it's our end. Thank you for watching and listening. Allah Hafiz.